I'm going to show you how to do a meta-analysis of correlations with study level corrections for artifacts. That is, for most, nearly all of the studies, you have information on the reliability of X, reliability of Y, and possibly on range restriction. Now, I'm assuming that you've watched the first video and you've already installed the software and it's in a directory off the C drive, meta-analysis programs that's set up by default by the program. I'm going to start the program in a second, but I want to show you what the data looks like. So I'm assuming that this is my coding sheet. So I've recorded a number of studies. I have the correlation. I have a sample size for each of those. And if you notice here, I have information on the reliability of my predictor, the reliability of the dependent variable, and I also have some level, some measure of range restriction. And I'll talk a little bit about that as we go along. I may have also coded multiple moderators. Here I just have categorical moderators, and these are the three that I actually coded. And then you'll see another column here, which is actually a copy of this file, and I'll explain to you how I did it. But this is kind of what my coding sheet looks like. So now I'm going to come over here. I'm going to launch the program by clicking on this icon. It has a couple of extras here. You can some templates for calculating biserial correlations, which you shouldn't need if you're aggregating correlations, but you may be interested in using the template for computing composite correlations. For example, if you had the relationship between job satisfaction and job performance, and you have 3 or 4 different measures of performance, as the textbook describes, it's advantageous to compute a composite correlation across those multiple dependent variables. There's the README file over here. As I strongly encourage you to read it. I would print it and have it in front of you as you do your first meta-analysis. But we're going to go ahead and get started. So as you see here, we're going to click here to start. And then we have a choice of doing 4 different types of meta-analyses. And as I said, in this one, we're going to be aggregating correlations, and specifically correlations where we have information on statistical artifacts at the study level. So I'm going to click on that. Now it immediately asks me if I have range restriction in the data. And if that data are available, I will click yes. And you can kind of read what it considers the problem of range restriction. Now, if I don't have range restriction information, I was unable to get it from the studies, or I'm not worried about range restriction. For example, most of my studies were very large samples. I would click no. I'm going to go through this, and I'm going to click yes here because it's a little bit more complicated. There's a good chance that you don't have range restriction in your data. You'll be able to follow this demo. It'll be a little bit easier if you click no here. But I'm going to go ahead and click yes. Now, the important thing to understand about the program is there's three steps involved in doing the meta-analysis. You have already created a coding sheet, and in that coding sheet you've recorded correlations and sample sizes for each of the studies. But that's your data, and it resides in a coding sheet that you've created. The program that doesn't yet understand or can read that particular data. So the first step is always entering the data into the program and then saving it, and then the program will go back and analyze the same data that you've recorded, that you've coded, but now it's integral or integrated into the program. So step 1 is getting the data in and saving it. Then step 2 is loading and analyzing that data, and that's a separate step. And then the third step is the easy one. That's writing up your analyses for publication. So we have not yet transferred our coded data into the program, so I'm going to click on Enter. And I have 2 different options for doing this. One is I can choose this particular option, and this will generate kind of a a access type interface for entering the data, and it looks something like this. And so what I could do is I can start plugging in my observed correlation, the sample size for that, my reliability estimates for X, my reliability estimate for Y. It's a little bit more reliable than that. And then my ratio, which could be a little u or a capital U, the estimator of range restriction. If I have moderators in the data, I would enter that here. I would have to enter levels. Say it's a categorical moderator, the smallest number I could list is a 1. I can't do, say, like if you're used to dummy coding, you can't do a 0 and 1, but you could indicate a 1 and a 2, or a 1, 2, or 3, where I'm indicating different levels of the moderator if I have that. And then, as you can see here, I can enter 8 on a page. I can click continue and continue entering individual studies. And if you have recorded or if you coded your data in a way where it's a little bit hodgepodge, this might be an easy way of organizing that data and getting in. But I'm going to show you what I think is an easier way. So instead of clicking on this, I'm going to click on Excel. And I keep doing this, and I'm not quite sure why. When you click on it, you'll pull up a data file that looks like this, and it should, as long as you don't save over that by clicking on this Save button, you shouldn't run into the problems that I just ran into. You'll also get a warning, a security warning. That's what the red text is here. And the first time you load it, depending on the security of your Excel program, there's a couple of blocks that the program puts on it, and then you'll have to click on options and disable the macros in order to be able to save the data. So this is now a template that the meta-analysis program has, and this is what it's going to analyze after you save the data. And it needs these columns here. Now, you'll notice that when we got started, I showed you the data set that came from my coding sheet. And you notice that it has the exact same columns here. And so my suggestion is that you recreate these columns. The headings don't exactly matter because you're only going to be copying and pasting data, but if you're working within your coding sheet and set it up exactly like this with study number, the correlation, the sample size, your U value for range restriction, reliability of X, reliability of Y, and something for a moderator, that it makes it so much easier to enter the data. Another point that I want to make is if you're missing data on any of these variables, you need to have the correlation and the sample size. But for range restriction or reliability, if you enter a 99, that value is read as a missing data point by the program. So I'm in my coding sheet. I have the columns to match the template in the program, and all I'm going to do is highlight the data. Come over one. And I'm going to copy that. I'm going to move back to the program template and paste it in. And as you can see, that saved me an awful lot of typing in that data entry program. And now all I want to do is save the data. So I'm going to click on Save Data. It's going to ask me for a file name, so I'm going to call this correlation sample. And you can see that it's off the root directory. There's a subdirectory for data 1, and that sample will be saved under there. I click Save. It's doing something. That tells me it's working. And when it stops moving, it kind of sits there, and I'm done. Notice here, if you have no range restriction in your data, you can enter 1. I should have mentioned that earlier, but I have range restriction in my data. I can just exit out of this. I'm done in here. And now I'm going to go back to the program, and it brings me back to this screen that we saw before. Last time we chose Enter, and remember there's the 3 steps. The first step is getting the data in and saving it. We've done that. And now we need to go and analyze the data. So we're going to reload the data that we just saved. So I'm going to click on Load. And now I will see all of the data sets available to me. These are all in that Data1 subdirectory. I'm going to choose Core Sample. That's what we just created. But I'm going to show you two things down here. If you're interested in exploring publication bias, a handy tool for that is the cumulative meta-analysis. And as the textbook points out, what that does is it orders the studies in this particular sample on the basis of, in this data set on the basis of sample size, does a meta-analysis with the largest sample, then a meta-analysis the two largest, and then a meta-analysis the three largest, and so forth. And the idea is that most unpublished studies are probably among the lower end, and what you're interested in is as you get to the lower sample sizes are the inclusion of those particular studies changing your estimates of the mean correlation, population variance, and so forth. The other option here is a moderator analysis, and I'm going to come back and show you that in a few minutes. But right now I'm just going to do my straight meta-analysis. It's very simple. As I'm reading the directions here, click the red button before the name of each data set to load it. So I'm loading this thing that I just saved. And now I saw the same screen that I did before. Remember, I was messing around up here. Now I'm going to come to the hand on the calculator to start analyzing the data. And now remember that I told it I had range restriction. And in fact, if you remember the data set, I had that column with the values of U. And now I need to tell it whether I have direct range restriction or indirect range restriction, because the calculation formulas vary a little bit. If you remember, on direct range restriction, my sample is restricted by selecting on the variable x that's correlated with y. And in the case of indirect range restriction, the sample is still restricted. I have a subset of the population, but they were selected on some variable other than x. And so for purposes of this demonstration, it doesn't really matter. I'm going to choose direct range restriction. It's going to ask me to name my output file, and I'm going to call that demo sample, or I'm going to call it demo output. And now it's asking me for a title. The difference between these two is this is a file name, and this will actually appear in the output. And imagine if you're printing the output several times or several different runs, you may want to have something right in the output to tell you what it is. So I would say something like main analyses, no moderator. And I'm going to hit enter, and now it says press any key to continue, and boom, it ran the analysis. So down here you see the bare bones meta-analysis. I have a mean R of 0.28. I have a corrected R of 0.55. That's correcting for both criterion and predictor unreliability. I can see here that 71% of the observed variance is due to artifacts. So I may potentially have a moderator in there, and I can now print. If I hit print, it's not just printing this, but it's actually printing all the results, and I want to show you what that looks like. So I'm going to just click here to get out of this. I've never saved my data. I think I might want to save the data if I'm modifying it, and I'll show you why I may want to modify in a minute. But now I'm back here. So let's go out of here for just a moment. Go back, and in our output file, our output directory under the main meta-analysis program, we have the demo output, or what did I call it? I forget which one I just called it. These should almost all be the same. It looks like it's this one. And so you can see what that looks like. It's telling me I had 24 studies, the mean R uncorrected, the corrected R, lots and lots of results. And then I can click on this and I can print that, or I can save it under a different file name. So that's that. So let's come back here. Let's go into the program, show you one more thing. I'm going to go back in. I'm going to do another meta-analysis. I'm going to again indicate that I have range restriction in the data. I want to load the data set again. And now what I want to look for, remember that there was a possibility of a moderator based on the 75% rule. If I want to do a main analysis, I simply click on the red button and get going. If I want to do a moderator analysis, I need to indicate that first by clicking on moderator, then choosing my sample. So I don't do sample run, I do click moderator analysis sample. I again indicate that I have range restriction in the data, and boom, it's already done that. You saw it run, calculation complete, and it shows me here where the output has been saved. You can view summary results, but there's not a whole lot particularly, I'll show you. This is all you get. It just shows you the mean R and standard error within each level of the moderator. Not particularly interesting, but I can come back here. I have 3 different output files. The output M is for the moderator. And what I see here is this is all of the results for the first level of my moderator. Here I see a mean of 0.31 and a row of 0.56, and I can see the estimated population. This is the standard deviation of the population row, and I can get the same data. You notice that the uncorrected R's are fairly different, but the rows are approximately equal. But you'll also notice here that I'm accounting for all of the variance in this level of the moderator with the statistical artifacts. I know that because this value is 0, and I also know it because percentage of artifacts is at least 100%. So that's how I detect moderators, and that is how to do a meta-analysis on correlation.